This video is on protein synthesis, specifically on transcription and mRNA processing. In protein synthesis, scientists over the years often talk about the central dogma. The central dogma is the idea that the genetic code is written in the DNA so that whenever we copy our DNA through DNA replication and we go through cell division, our new cells all have a copy of that DNA because that genetic code is essentially our genotype, our gene combination. And if you remember genotype, we're talking about big T, little t, etc. Okay. And our genotype is then expressed as a trait through this process of protein synthesis. So it was the idea that, hey, first we need to copy the DNA into mRNA. And there's kind of two steps of that: transcription and mRNA processing. And then we get our mRNA. Okay, and then here's our nucleus. Of course, we're copying our DNA into mRNA, one, so we can highlight specific genes to copy, and two, because the DNA can't leave the nucleus. So then the mRNA for that specific gene that you've copied leaves the nucleus, and through translation and post-translational modification, you get a protein, right? And this protein helps to interact with other structures in your body, or maybe multiple groups of proteins to form a trait. So this is kind of like our phenotype here. And this is what's kind of known as the central dogma. You go from DNA to mRNA to protein to express this trait. Okay. And this overall process right here, from DNA to mRNA to protein, is known as protein synthesis. And sometimes we even call it gene expression. We know now from years of research that there's actually many times that we don't follow these steps in exactly this order, or many times where a trait is actually encoded for through many genes and many proteins interact to form that trait. Sometimes with viruses like HIV, they can go from RNA to DNA, back to RNA, back to proteins and things like this. But this is the central dogma, like the typical flow, although there are alterations of this flow and other things that can happen. And again, your DNA stays in the nucleus. The mRNA can go from the nucleus over here to like the ribosome, cytoplasm, that kind of situation. So today we're going to focus on transcription and mRNA processing. Transcription occurs in the nucleus because transcription is the process where we copy specific genes in the DNA into pre-mRNA. And because we're copying DNA into mRNA, it has to happen in the nucleus because DNA can't leave the nucleus. So let's start with our definition of transcription. Transcription, like I said, is when we cut specific genes. I shouldn't say cut, copy. When we copy, not cut, copy specific genes into pre-mRNA. And the process of transcription has three main steps: initiation, elongation, and termination, which I'll abbreviate as I, E, and T when I'm writing on this diagram. Okay? Like I said, it occurs in the nucleus. So what happens during transcription? The first step is initiation. And then we have elongation and we have termination. In initiation, you have specific proteins called transcription factors. Transcription factors right here are proteins that highlight the specific gene to be copied. So they find a region on the DNA, okay, called the promoter region. This promoter region is a section of the DNA that kind of like marks the start of specific genes. So these transcription factors or proteins find this promoter region, the promoter region often has what's called a Tata box, and they bind to that promoter region on the DNA. That's then the signal for an enzyme called RNA polymerase to attach. RNA polymerase, much like DNA polymerase makes DNA, we have our RNA polymerase. RNA polymerase attaches to the DNA at the promoter site on the DNA because of the transcription factors which tell it where to attach. This is the first step of transcription called initiation. Next, there are two sides of the DNA. RNA polymerase does not travel down both sides of the DNA. Okay, the two strands of DNA, one is called the template strand and one is called the coding strand. What's going to happen is the RNA polymerase is going to travel down the template strand on the DNA, right? So this section right in here, okay. This here is one side of the DNA. And this here is the other side of the DNA. Okay. Called the coding strand. RNA polymerase travels down the template strand on the DNA, and it pulls in RNA nucleotides that are just kind of hanging out in the nucleoplasm, in the fluid inside the nucleus. And it does complementary base pairing DNA to RNA. So here's our DNA. RNA polymerase is going to move down, and it's going to pair RNA nucleotides with DNA nucleotides. And this is going to create our pre-mRNA. Etc. Okay, so this is our pre-mRNA that's going to be made. This step of transcription is called elongation because we're elongating. The RNA polymerase is reading the DNA, bringing in RNA nucleotides, but covalently binding those RNA nucleotides together to make that section of pre-mRNA. Okay. So that's the second step of transcription. We're making our mRNA. And you'll notice it looks very similar to the DNA. However, of course, RNA nucleotides use ribose sugar instead of deoxyribose sugar. Okay. And instead of using the nucleotide thymine, they use the nucleotide uracil. So the DNA, if the DNA codes for an A, the RNA will code for a U. Okay. So basically T's are replaced by U's in the RNA. So elongation, we're making our mRNA strand, and then we get to a termination sequence on the DNA. So we get to a termination sequence. This termination sequence on the DNA basically tells RNA polymerase, all right, you're done. You have made the entire pre-mRNA, and RNA polymerase can detach, okay, and be reused again. Detaches, it can be used again, okay, and now you've made your mRNA, your pre-mRNA, the pre-mRNA detaches, right? The DNA goes back together, and that process of transcription is over. So this is called termination. And if you flip your big sheet over, you can actually see kind of in more detail the steps of transcription, right? All of this picture is essentially showing you initiation. Right? And you can see the RNA polymerase binds to the DNA at a specific promoter region. Okay, so the promoter is a marker before a gene. So here's the promoter region right here. It marks the beginning of the gene. Okay, it's in the DNA. After the promoter region, then you actually find the gene that you're trying to copy into mRNA. So your transcription factors, like I said earlier, your transcription factors come along to mark where the RNA polymerase should attach. So your transcription factors mark where the RNA polymerase should attach. Okay. Then based on this marking, you have general and specific transcription factors. The RNA polymerase actually attaches. So you can kind of see all the pieces here. This is the promoter region on the DNA, which told the transcription factors, hey, you should attach here. The transcription factors then told RNA polymerase to attach. Now that RNA polymerase is attached, it can move down this DNA. So here's the DNA. Okay. All of this step, once RNA polymerase is attaching, is the initiation step. So initiation is really the first step. In elongation, we have elongation right here. The RNA polymerase attaches RNA nucleotides to the 3' end of the growing strand. So right here, that's elongation. We're going to make the mRNA here. Okay. And then finally, we get to the end of the gene, and we have termination, which is the end. We get to the end of the gene, and the process stops. Like I said, the DNA goes back together, the mRNA can leave, and the RNA polymerase can be reused. So that's transcription. Why do I keep calling it pre-mRNA? I keep calling it pre-mRNA because the next process is called mRNA processing. I'm actually going to stop this video here and at transcription, and I will make a second video on mRNA processing.